Hello, everyone. Welcome to Antibody Engineering and Therapeutics Digital Week, brought to you by the producers of the face-to-face antibody events. My name is Michael Keenan. I'll be your host today for the session. First, I'd like to cover some quick housekeeping items. If you experience difficulties with audio or advancing slides, refresh your screen with F5. That's F as in Frank, number 5. If you are experiencing other issues, just hit the question mark button to receive assistance. At any time during the presentation, submit your questions into the Q&A window on the left-hand side of your screen. In 24 hours following this presentation, you will receive a link to watch the recording of this session. You can also download a featured white paper in the resource list box on the right side of your screen. Let's go ahead now and introduce our speakers. We have Shireen Khan, who's the Senior Director of Biologics at ChemPartner, and Dina Siripano, who's Senior Technical Sales Specialist at Berkeley Lights. And their presentation title today is Rapid Discovery of a Diverse Panel of Antibodies Against Membrane Targets Using the Beacon System. Thank you for joining us today, Shireen and Dina. Now I'll hand it over to you to begin the presentation.
Thank you. Today we're going to talk about discovery of a diverse panel of antibodies against a membrane target using the Beacon platform. So first I thought I'd talk about the different antibody discovery strategies that are commonly used, including phage display, hybridoma, and B-cell cloning. So each of these methods has their pros and cons. Of course, for phage display, the library is already generated, and it's just a matter of bringing the antigens forward to do the panning campaign. There's flexibility in terms of the type of antigens that are possible, but it is challenging for multi-path transmembrane proteins such as GPCRs and ion channels. And another challenge is that you may need to affinity mature the candidate. The screening format is typically scFv or Fab for phage display, so there is some risk in reformatting to IgG, but together it's a beneficial platform for accelerated discovery as well as antigen flexibility. For the animal immunization-based technologies, including hybridoma and B-cell cloning, the advantages are that sort of in vivo affinity maturation that can take place in the animal. For hybridoma, it's a much longer cycle time of about 6 to 8 months, and the total number of hybridoma candidates that you can typically screen through is on the order of 5 to 10,000 members. But as we're aware, there's challenges for toxic, conserved, or less immunogenic targets. But there's been quite a bit of success with challenging target classes, including GPCRs and ion channels, because of the ability to use genetic immunization or cells overexpressing targets of interest. The advantage, again, being perhaps not needing affinity maturation and using the IgG format straight away, but we really see B-cell cloning as an advantage, combining hybridoma as well as phage display advantages in the sense that you likely do not need to do affinity maturation. You potentially get greater sampling of the immune repertoire with potentially tens of thousands of single B cells evaluated from a single immunization campaign, and accelerated discovery. But probably the most important aspect in our minds is the ability to move function forward in the antibody discovery process, which we'll go through in more detail. So at ChemPartner, we are doing B-cell cloning on the Berkeley Lights Beacon platform. And the Beacon platform itself enables the evaluation on multiple optoselect chips. So up to 4 chips can be loaded onto the instrument. And on these chips, we can actually position single B cells onto the chips that have thousands of these sub-nanoliter chambers called nanopens. And we use controlled patterns of light to be able to move these single B cells, to cluster them into these nanopens, allow them to secrete antibodies so that we can assay for target-specific binding as well as function. So as I mentioned earlier, the value proposition really is in being able to move function forward in the drug discovery process. So the Beacon platform enables us to do assays either in the channel or within the pens. So what's shown on the left here is a nice bloom of bead-based binding forming above the pen that has the B cell secreting that target-specific antibody. And it's possible to actually do these assays with cells as well. So in addition to binding, you can do cross-reactive binding. And some of the additional assays that we're very interested in developing include cytokine release as well as epitope binning, gene expression assays, as well as cytotoxicity on the platform. Another set of assays that we've actually developed on the platform that have been quite helpful in selecting lead candidates include both cell as well as bead-based assays, binding assays on the platform. So this is done by loading the cells overexpressing, in this case, human PD-L1 into the pens, as well as importing in beads that are coated with human PD-L1. And so this enables us to visualize the bead-based binding within the channel as a bloom, as well as signal within the pen. And in addition to that, we're able to assay for target-specific blocking activity. And so what we did in this case was use soluble PD-1 that's fluorescently labeled with Electrofluor 488. And by importing that in and looking for whether there's signal in the Texas red channel, which is actually our anti-mouse IgG secondary detection antibody, as well as the FITC signal, which should be the PD-1 fluorescently labeled, we can evaluate and categorize our antibodies as far as whether there's specific binding to beads, cells, or whether the presence of that antibody prevented the association of the fluorescently labeled PD-1. So you can see at the bottom there where there's signal in both the Texas red as well as the FITC channels, you're getting basically an antibody that's able to bind, but it's non-blocking activity, allowing PD-1 to bind. Whereas at the top, you're seeing specific binding to the beads in the cells, but exclusion of the PD-1 signal, suggesting that that's a binding, not blocking antibody as well. So what we've also spent quite a bit of time on is evaluating cell-based binding activity. The advantage of being able to do cell-based binding in the channel is that you get to see with either engineered cells that overexpress your target of interest or endogenous cells that express high enough levels of your target so that you can actually evaluate the number of hits that you get across an entire chip by using the Beacon software. So that's actually a very important number to kind of get an idea of across an entire chip what the hit rate is. And so we've actually been able to evaluate cell-based binding across a variety of cell types and confirm that that binding is localized to a particular pen by also loading cells into those pens and looking for cell-based binding within the pen as well as within the channel. And so the case study that we're going to cover today is actually a collaboration project with Charles Craig's lab at UCSF and Shing Pharma Innovations. And the focus was a membrane-bound target that's implicated in triple-negative breast cancer. So triple-negative breast cancer is a type of metastatic breast cancer, which is the most advanced stage of breast cancer. And oftentimes in metastatic breast cancer, the disease spreads to the bones, lung, or liver. This represents a very high unmet medical need as there are many patients suffering from the disease and no therapies thus far have been completely curative. And there's over 150,000 patients in the U.S. Triple-negative breast cancer in particular accounts for about 10 to 20% of all cases of breast cancer, and it's characterized by low expression of progesterone, estrogen, as well as HER2 receptors. And so conventional treatments such as hormonal or immunotherapy that are commonly used for metastatic breast cancer tend to not be effective for triple-negative breast cancer. So in Charlie's lab, they've demonstrated proof of concept both in vitro as well as in vivo for targeting a particular membrane-bound protein called the urokinase plasminogen activator receptor, or uPAR. And with their data have really demonstrated nicely the potential for having anti-uPAR antibodies as an anti-cancer therapy for triple-negative breast cancer. So they've reported using triple-negative breast cancer samples that there is a statistically significant increase in uPAR mRNA level in these patient samples compared to all other breast cancer subtypes. They went on to look at different breast cancer cell lines and found again that there was a correlation between an increase in uPAR gene expression in breast cancer cell lines that have the triple-negative breast cancer signature compared to human mammary epithelial cells. And so at the gene expression level, there's higher amounts of uPAR, and that also correlated with higher levels of the uPAR protein on the surface of the triple-negative breast cancer cells, including MDA-MB231, shown at the bottom right here. And so the normal role for uPAR in cells is to actually promote both extracellular functional outcomes as well as intracellular functional outcomes. So in particular, uPAR is a GPI-anchored membrane protein that associates with uPA to promote ECM degradation as well as activation of MMPs and proliferative signaling. uPAR also interacts with integrin family members, including alpha-5 beta-1 and alpha-5 beta-3. And by associating with these integrins, it promotes intracellular functional outcomes, including cytoskeletal changes, proliferative responses, as well as cell migration. So you can imagine, just looking at the normal function of uPAR, that having the ability to localize the ECM degradation at the surface of a cancer cell can enable a tumor cell to invade surrounding tissue and potentially intravasate into the circulatory system, extravasate, and promote metastatic seeding. So in the context of cancer, uPAR functional activity can promote tumor growth, invasion, as well as progression, and uPAR expression is often associated with more aggressive types of cancer. So in Charlie's lab, they've been very interested in generating anti-uPAR antibodies with inhibitory activity. So they initiated a phage display panning campaign and identified 12 antibodies that bound to human but not cyno or mouse orthologs of uPAR. And two of the antibodies, namely 2G10 and 3C6, were developed further by his lab. 2G10, as shown in the schematic, is an inhibitor of the uPA-uPAR association, whereas 3C6 inhibits the association of uPAR with integrins. And so in his lab, they went on to publish that the two antibodies, 3C6 and 2G10, have the ability to inhibit phospho-ERK levels in triple-negative breast cancer cell line MDA-MB231. So on the upper right there is showing that 3C6 and 2G10 can inhibit phospho-ERK induction quite effectively. And in the lower right, they're able to show that both of these antibodies also inhibit invasion through a cross-linked matrix in, again, the MDA-MB231 cells. So the next approach was to evaluate whether these antibodies that had the in vitro activity could also limit tumor growth in vivo. So they looked at the MDA-MB231 xenograft model and dosed with both 3C6 and 2G10 at 30 mg per kg, and showed that particularly for 2G10, it really blocked tumor growth, and 3C6 slowed the tumor growth, which demonstrated that very nice inhibitory activity in vivo. They also evaluated 2G10 conjugated to a therapeutic radioisotope, which showed tumor regression. And this kind of gave a little bit of hint that perhaps inhibiting uPAR alone would not be sufficient to induce tumor regression, and you might need an additional cytotoxic effect on top of inhibiting uPAR signaling. So this was very promising in vivo data suggesting that targeting the uPAR pathway, not only in vitro but also in vivo, caused anti-tumor effects. But the one challenge with this particular approach is that the antibodies bound with high affinity to human uPAR, but there was no binding to either mouse or cyno uPAR. So if there was interest in developing the candidates further for safety or tox studies, we'd really need to have that cross-reactive binding. So that's where we came into the project and brought forth the capabilities of the Beacon platform to really screen deeper into the immune repertoire to identify cross-reactive anti-uPAR antibodies, which were difficult to identify thus far. So our goals were to seek human as well as cyno cross-reactive antibodies, whereas mouse binding would be a nice to have, but not necessarily a must have. So we developed multiplex binding assays on the Beacon to identify species cross-reactivity upfront using a bead-based capture assay of the recombinant proteins that were differentially labeled. We would then export the B cells that secreted the antibodies with these desired binding profiles. We would sequence them, express, purify, and then characterize based on the target-specific binding and functional properties of uPAR, and this was done with UCSF. And so ChemPartner was uniquely positioned to be able to bring forward a lot of the functional areas within our therapeutic antibody discovery platform. So in particular, reagent generation was used for this project to generate the cyno uPAR recombinant protein. And we also leveraged our capabilities in our protein sciences group to generate the recombinant antibodies. We developed assays for this project, binding as well as functional assays. This was all done in Shanghai. And then the antibody generation was actually done in our South San Francisco location for the B-cell cloning effort. The VHVL sequencing was done in Shanghai as well as the manufacturability assessment. So we were able to bring forward an array of capabilities to support this project. So this is the sort of distribution of effort for the uPAR project. Of course, the UCSF team is highly skilled in doing the immunizations. So they took the SGL strain and immunized with human uPAR that they generated in the lab of Charlie Craik. And once the animals were ready for takedown, we actually did the isolations of the B cells in our South San Francisco location, put them onto the Beacon and ran the multiplex assay, exported the B cells. The cloning, expression, purification, as well as the sequencing happened in Shanghai. And then further characterization will be done by both the UCSF as well as the ChemPartner team with FAX binding, phospho-ERK, and ADCC done in Shanghai, and ELISA binding, looking for and confirming cross-reactive binding by ELISA, as well as the blocking of uPAR, affinity assessments by Octet, and invasion assays all done by UCSF. And all of this data taken together would be used to select 2 to 3 top antibodies to move them forward into an orthotopic breast cancer model, specifically triple-negative breast cancer. So the ChemPartner B-cell cloning workflow is as shown here. So while the UCSF team was immunizing the SGL animals, which were 3 groups of 3 mice per group, we were generating the fluorescently labeled antigens, QCing them by FAX, and evaluating them on the Beacon for robustness of the signal. And once it was ready for takedown, we harvested the spleen and bone marrow and did the isolations using CD138 positive isolation kits. We penned them onto a 14K chip and ran the multiplex assay, exported the hits with desired binding profiles, and then did single B cell sequencing. So this is what the multiplex in-channel binding assays looked like on the Beacon. So of course we loaded the plasma cells onto the chips, allowed them to secrete antibody, and then imported the anti-mouse IgG capture beads along with the differentially labeled human, mouse, or cyno uPAR proteins. And so we allowed this assay to develop so that we could start to see enrichment of signal, that is capture of the differentially labeled antigens on the surface of those beads. And so here is representative images of this multiplexing assay. So we have the human uPAR proteins shown as image 0 versus image 11. And this is a time lapse of about 45 minutes where we start to see between image 0 and 11 that increase in signal on the beads. So enrichment of signal suggesting that the antibodies were recognizing human uPAR. And then on the right-hand side, we're showing that across that same time course, there's also an accumulation of signal suggesting that the antibodies are also recognizing cyno uPAR. Now, in this study, we were not able to see binding to the mouse uPAR protein, but again, that was a nice to have, and really cyno binding was the must-have. And so the outcome of our B-cell cloning campaign, we ended up evaluating over 41,000 single B cells on the platform. We ended up seeing a lot more human-only binders, but we did see at least 79 human cyno cross-reactive binders during the screen. So the hit rate is about 0.2%. And we further narrowed down the 79 antibodies by looking at how many of those had a single B cell in the pen. And so a majority of them did. It was 64 total. And so we went ahead and recovered the cDNA from those 64 single B cells. And the recovery of complete sequences was actually quite high. So we were able to pull out 50 that had good sequences. So the recovery rate was about 78%. And 100% of those were sequence unique. So 8 of them shared the same VH sequence and 6 of them shared the same VL sequences, but once you paired the VH-VL sequences together, all of them were unique. And so in this slide here, you can see the hierarchical clustering tree of all of the antibodies that we pulled out, suggesting that they are unique. And buried in here are the 2G10 as well as the 3C6 antibodies. So a very diverse panel was pulled out from this B-cell cloning effort. The next evaluation we did was a hotspot analysis. So the advantage of having the sequence so early in the drug discovery process is that you can, earlier in the process, do the manufacturability assessments and look for specific hotspots within the CDRs as well as the frameworks. So this enabled us to really kind of focus in on the most promising clones, but also do a retrospective analysis to really assess the predictive value of an analysis like that early on. And so shown in the graph here is the frequency of each of these liabilities and then the frequency of issues with either purity or expression. And so not surprisingly, what we found was that having an unpaired cysteine in the light chain correlated most highly with issues in either purity or expression. And so after doing all of these analyses, we found that out of the 50 unique antibodies that we had, 40 of them expressed at sufficient quantities in order for us to move forward and do functional assessments, including FACS binding using the MDA MB231 cells as well as ADCC. Okay, so this is representative data of the binding of the uPAR, anti-uPAR antibodies to the MDA MD231 cells. And in red is the 2G10 benchmark antibody. So you can see there's several candidates here that have pretty good binding by FACS, equivalent to 2G10 or even left shifted, meaning a stronger binding, lower EC50 values compared to the 2G10 antibody. And the next assay that we did was to evaluate the antibody-dependent cellular cytotoxicity. So going back to that earlier study done in Charlie Craig's lab where they conjugated 2G10 to the therapeutic radioisotope, suggested that you probably need a little bit more than just inhibition of uPAR in order to get a really robust anti-tumor response. So there was definitely interest in ADCC activity. So this assay was developed in Shanghai, and we used MDA MB231 cells as well as an NK cell line overexpressing the Fc gamma R3 receptor, and evaluated the 2G10 antibody, which showed about 40% cytotoxicity and an EC50 of about 11 nanomolar. So there was ADCC activity for this antibody, and that set us up quite nicely to evaluate our B-cell cloning antibodies, and kind of overlooked this particular detail, but all of these antibodies were generated as a chimeric antibody at this point. And so this assay demonstrated that compared to 2G10, there were a few antibodies that had strong binding activity, but also had fairly good ADCC activity. So the top cytotoxicity for some of them was a little bit higher than 2G10, particularly for 13706. You can see a top cytotoxicity percentage of almost 50% at an EC50 of about 5 nanomolar. So several candidates came forward that looked like they had fairly good ADCC activity and good binding properties. And the results are summarized here in this table. And these are sorted actually based on FACS binding results. So the binding EC50 values, the lower the value, the stronger the binding profiles. And you can kind of start to see highlighted in yellow here that the strongest FACS binders did not necessarily have the greatest ADCC activity. So we went ahead and looked back at the hierarchical clustering tree so we could kind of get a sense of which antibodies fit into each of these different categories. And in fact, what we found is that the antibodies that had the greatest, strongest binding profiles and the lowest EC50s clustered in one region of the clustering tree, and those with the best ADCC activity clustered in a completely different area of the clustering tree, and those binding EC50s tended to be higher. So interestingly, this suggests that there might be functional epitopes on uPAR that confer particular functional outcomes, and this needs further investigation. So in summary, we were able to evaluate over 40,000 single B cells on the Beacon platform. The hit rate was about 0.2%. We got a high rate of recovery of very good quality cDNA, and 100% of the clones, that's 50 total, that were sequence unique, had binding to human and cyno uPAR proteins. So even though some of the clones shared the same VH or VL sequences, all of them were unique. And so of these 50 unique antibodies, 40 of them expressed well in the first attempt, and we were able to do retrospective analyses of the manufacturability assessment and really select the most promising candidates to move forward with. We identified strong binders that had ADCC activity greater than our benchmark antibody 2G10, and based on the Beacon assays, look to be cross-reactive. So characterization of these antibodies is in process, including confirmation that We do have cross-reactive binding by ELISA. We're also planning to do uPAR blocking activity, affinity assessments, as well as functional assays, including signaling and invasion assays to confirm inhibitory activity. So with that, I'll just acknowledge the team at UCSF as well as ChemPartner for executing all of the studies that were shown in this presentation, and thank you for your attention.
Thank you, Shireen. My name is Dina Serpeño, and I'm the Senior Technical Sales Specialist at Berkeley Lights. Now I'm going to expand on how it's possible to select better lead therapeutic candidates in less than a week using the Opto-Plasma B Discovery 2.0 workflow for rapid, specific, and functional screening assays. The B-cell repertoire is vastly diverse, containing a network of billions of antibody molecules that each have a unique sequence and function. These diverse libraries of antibody molecules are ripe with therapeutic candidates. Over the last 3 decades, we have developed antibody therapies for a host of different diseases, ranging from autoimmune disorders, infectious diseases, and cancer. Despite their success to date, traditional antibody discovery technologies require a huge amount of time and resources to screen specific and functional antibodies. As a result, antibody therapies have only been developed against simple targets with limited to no success against far more difficult and complex membrane targets like GPCRs and ion channels. The discovery of antibody therapies against these difficult targets are going to require new technologies that can perform rapid, specific, and functional screens of the entire B-cell repertoire to access the high-hanging fruit. In this presentation, I'm going to demonstrate how Opto-Plasma B Discovery 2.0 on the Berkeley Lights Beacon system can accelerate the selection of better lead candidates in under 1 week. Antibody discovery using the traditional hybridoma approach requires immortalization of B cells by cell fusion, followed by a very lengthy process of expansion, subcloning, and screening. And the immortalization step often results in the death of more than 90% of B cells, which is a significant loss of B cell diversity for screening and selection. The Beacon platform bypasses these inefficient hybridoma fusions by directly screening plasma B cells from immunized animals. The plasma B cells are harvested and purified from diverse B cell compartments, including the spleen, bone marrow, and lymph nodes. These cells are then functionally screened on the Beacon platform, and antigen-specific sequences are recovered in less than 1 week. CLI's unique nanopen chambers and OEP technology enable this rapid, precise, and highly sensitive assay screening. In minutes, secreted antibodies from the plasma B cells can be assayed to determine antigen specificity. In this assay, the target antigen is conjugated to beads and mixed with a secondary antibody prior to importing into the chip. Secreted antibodies from plasma B cells diffuse out of the pen, bind to the antigen-coated beads, and then a secondary antibody is then sequestered on the bead to provide a rapid fluorescent readout in a matter of minutes. Using our nanopen technology, along with multiple runs of the Opto-Plasma B Discovery 2.0 workflows on the same plasma B cell sample, you can increase your screening throughput. In the workflows depicted in this figure, plasma B cells were screened on the initial day of harvest and purification, day 0, and the remaining sample was screened on the next day, day 1, after overnight tissue culture in Berkeley Lights' proprietary plasma B cell media. The plasma B cells that are subjected to freeze-thaw cycles also yielded hits, but with reduced hit rates due to lower cell viability. But by running multiple workflows on the same plasma B cell samples in our validation studies, we've demonstrated that you can discover greater than 5,000 hits per sample, if not more, depending on your studies. Here's a case study where the analysis of thousands of hits pose a major challenge for downstream characterization and selection using standard cloning and reexpression. We have demonstrated the ability to down-select lead candidates by performing multiple assays to functionally characterize antibodies. We performed a case study to demonstrate the down-selection of lead candidates using PD-1, PD-L1 model system. In this case study, we screened plasma cells that were immunized from immunized mice with PD-L1 using 3 assays. First, a bead binding assay to determine antibody specificity to recombinant PD-L1. Second, a cell binding assay to determine antibody specificity against a cell surface PD-L1. And then last, an assay to identify antibodies that block the PD-L1 receptor ligand interaction. The blocking assay is performed by co-incubating plasma B cells with PD-L1 expressing cells in the same nanopens. Blocking antibodies bind the surface of reporter cells, preventing binding of the fluorescent labeled PD-1. So pens with non-fluorescent reporter cells contain plasma B cells that are secreting blocking antibodies. In this study, we screened over 30,000 single plasma B cells and discovered nearly 600 hits. However, less than half of these antibodies bound the cell surface of PD-L1, and only 46 antibodies actually blocked the PD-1-PD-L1 interaction. The ability to down select these lead candidates using multiple assays eliminated the need to recover, sequence, clone, re-express, purify, and characterize over 500 irrelevant antibodies that did not exhibit the desired function. So this allows us to focus on only the most relevant, specific, and functional antibodies of interest. Of all the hits, and 0.1% of the screened plasma B cell repertoire, the lead candidates were only 7%, demonstrating that we can find these exceptionally rare functional blocking antibodies. Optoplasma B discovery can screen plasma B cells from multiple organ compartments. We've demonstrated that lead candidates are more abundant in bone marrow than spleen. These lead candidates may be missed otherwise by traditional hybridoma technology, which is typically performed only with mouse splenocytes. We sequenced the blocking antibodies and confirmed that these lead candidates were indeed unique to previously commercialized anti-PD-L1 antibodies, showing that we can still uncover a vast diversity of therapeutic potential that has not yet been tapped into. We cloned and re-expressed 24 of those blocking antibodies and demonstrated that these lead candidates also bind the native antigen, using ELISA to confirm binding to the ECD extracellular domain, as well as FACT to confirm binding on the surface of PD-L1 expressing CHO cells. We also demonstrated that these lead candidates bind the cynomolgus monkey species variant of PD-L1, which is critical for preclinical animal studies. Finally, we confirmed that these lead candidates do indeed have functional ligand receptor blocking activity using conventional well plate-based assays, where at least 5 of those antibodies had IC50 values comparable to therapeutic antibodies, and a couple with even higher affinity at the sub-nanomolar level. Now expanding on functional assays, in another case study with respect to viral neutralization, which is most urgent to our current pandemic situation, is the ability to search for blocking antibodies against SARS-CoV-2, a viral spike protein. It's a known target site to bind to the human ACE2 receptors in the respiratory airway, and only a fraction of antibodies that bind the viral spike protein will actually block and neutralize the SARS-CoV-2. The animation here represents an on-chip assay that distinguishes antibodies that functionally block the binding of SARS-CoV-2 RBD, receptor binding domain, to the human ACE2 receptor. In this assay, B cells are co-localized with beads conjugated with the spike protein RBD, and the blocking antibodies that bind RBD and prevent binding of fluorescently labeled ACE2 molecule. Therefore, nanopens in which PET beads are not fluorescent contain B cells that are producing antibodies that may block SARS-CoV-2 infection of human cells. We can identify antiviral antibodies from purified human B cells using convalescent patient samples. The full-length viral proteins or their subunits can be conjugated to the beads, which we then flow into the channels along the top of the nanopen containing the plasma cells. And using a fluorescently labeled anti-human secondary antibody, we can detect the secreted antigen-specific antibodies within minutes. In these images, we identified antibodies specific to the receptor binding domain, also known as RBD, and see if there's cross-reactivity to the full-length spike protein of the SARS-CoV-2 virus. This work was performed in collaboration with our partners at Vanderbilt University Medical Center. The images here represent a workflow in which 3 assays were performed to select antibodies that bind the receptor binding domain, or RBD, of the SARS-CoV-2 spike protein and blocking binding of the RBD to the human ACE2 receptor. Assays 1 and 2 were used to confirm antibodies that bound RBD on beads in the channel and also in pen, respectively. Then in assay 3, we took a look at the blocking capability of our antibody between RBD and ACE2. In this top row of images, we see nanopens in which a single B cell secreting antibody that binds RBD conjugated beads, but that did not block the binding of the beads to the fluorescence ACE2 molecule. In the bottom row of images, we identified a B cell secreting antibodies here that both bind RBD and also block binding to ACE2, as confirmed by the absence of expression of fluorescence on the RBD-coated beads. Using viral binding and blocking assays in a single workflow, we're able to rapidly select the 10 rare RBD ACE2 blocking antibodies out of the nearly 900 RBD binding antibodies. The identification of blocking antibodies during the primary B cell screen reduces the need to sequence, clone, and re-express over 800 irrelevant antibodies that don't exhibit desired blocking activity. Rapid selection of blocking antibodies can accelerate the down selection of lead candidates for SARS-CoV-2 antibody therapies. In-vitro neutralization assays are also key to selecting antiviral lead candidates. We've demonstrated a proof of concept here with a viral neutralization assay using a vesicular stomatitis virus, VSV, pseudovirus that was engineered to express the Ebola glycoprotein. We created a set of hex cell lines secreting either neutralizing or non-neutralizing antibodies and mixed these together. We then loaded the single hex cells with vero cells into individual nanopens. We used our standard bead-based assay to confirm that the hex cells were secreting antibodies. So simultaneously, we identified pens in which antibodies blocked viral infection, as well as confirmed by the absence of expression of a fluorescent GFP marker in the vero cells. This assay demonstrates that we can successfully identify cells that are secreting viral-neutralizing antibodies. Not only can we screen for these specific and functional antibody hits, we can also recover them. OptoSeq BCR is a specialized workflow that enables recovery of antigen-specific antibody sequences from plasma B cells with high efficiency by integrating on-chip genomics. In our validation studies, we recovered paired heavy and light chain antibody sequences from an average of 86% of plasma B cells across multiple workflows and multiple plasma B cell samples. This high sequence recovery efficiency was observed for both fresh and frozen plasma B cell samples. After the beads are recovered into a 96-well plate, antibody heavy and light chains are amplified for sequencing. In just 2 days, optoplasma B discovery can enable single plasma B cell cloning, screening, on-chip genomics, and sequencing of antigen-specific functional antibody sequences. By enabling functional screening of the B cell repertoire and automated sequence recovery using both OptoSeq BCR and the Optoplasma B Discovery 2.0, we can reach the high-hanging fruit for antibodies against difficult targets. Thank you.
Great. So thank you for that excellent presentation, Shireen and Dina. We've received a few questions already in the chat pod, but I'd like to remind everyone, if you have a question, feel free to type it in now, and we'll make every attempt to get that answered. So go ahead and do that now. I'd like to thank Berkeley Lights for sponsoring today's event, and also just wanted to remind everyone to check out the resources list to the right of your screen, where you can download a few featured white papers. So let's get back to Shireen and Dina and begin the Q&A. And I will go ahead and read the questions for everyone, and we will go from there. So it looks like the first question we have is a question for Shireen. What was the real time required to screen the 40K B cells?
That's a great question. So the way the study was done, it was actually a staggered approach. So we did immunizations staggered by about 2 weeks each over the course of three total runs. And so each of them were actually done, and we focused on basically 1 week to do the analysis. So the takedown and the loading onto the beacon happened in 1 day, but there was actually potential because we've developed a media formulation where we can keep the B cells alive for over a week in the incubator. So if we need to get back onto the beacon within that same week with that material from the takedown, we can actually load onto the beacon and repeat assays as needed. So with that ability to keep them alive and continuously assay, we are able to do multiple runs within that week. And again, it's staggered by 2 weeks per run, so it allows us time to optimize the assays if needed so that we can kind of boost up our hit rate and robustness of the assays in that time frame.
Great. Thank you for that. Looks like we have another one for you, Shireen. This one says, thank you for your descriptions, very useful and helpful. When you were working in the pen, was a light trap used to keep the ASC in the pen, or was that gravity?
So it's interesting the way, and perhaps Dina can weigh in on this as well, but when you actually move the B cells into the nanopens, they stay in there. So when we're flowing in either continuous perfusion of media or introducing either beads or other reagents for assays, the B cells stay in the nanopen quite efficiently. So we didn't actually have to do anything to keep them sequestered in the pen. So that kind of works pretty well.
Yeah, and I can go ahead and add on to Shireen's great description there. Just based on the fluidic dynamics, anything that's flowing in the areas above the pen can move, but anything that's basically sheltered inside the pen will stay in the pen, and there's a slight 1 degree tilt that the chip sits at. So basically, by gravity, they do stay in there. That's correct. And so there isn't any movement of those cells outside beyond that.
Great. Dina, it looks like the next question's for you. Here it is. Now is COVID-19 time. Have you used your beacon technology to isolate anti-SARS-CoV-2 antibody clones from human B cells?
Yes, that's a great and timely question. So as you've seen, right, in our presentation, we've done a study with University of Vanderbilt Medical Center, as well as a few others that are part of now what we call the GPI consortium to address the SARS-CoV-2 pandemic. We are able to take plasma cells from convalescent patients. So we'll take those by going through, an enrichment process and then doing a differentiation and activation. And then once we get the antibody secreting cells, we can go ahead and go through the same process of loading it onto a chip in conjunction with doing all the different assay screening where you're looking for either specific binding to, you're looking at IgG secretion, you're looking at specific binding to either the RBD or the full-length protein. And then in addition to that, you can have reporter cells to see if there's any infectivity or blocking that's happening, the blocking of infectivity that's happening within the pen as well, as demonstrated in the slides that you've seen. So that's a great question.
Perfect. Okay, looks like the next one's for Shireen. How is it when you compare your beacon method with cell sorting method in terms of isolating single B cell?
Yeah, that's a great question. So we actually use magnetic isolation to enrich for the B cells before we go on to the beacon. So with the sorting method, you can actually basically achieve the exact same thing and sort the population of interest straight away. But we've found that the magnetic isolation has been best at preserving the viability of the cells, because essentially after you load onto the beacon and then run the multiple assays, you really kind of have to keep the B cells as happy as possible, and we find that the magnetic isolation is gentler on the cells prior to loading onto the beacon and then going through that longer workflow.
Great. Okay, thank you for that. Looks like we have another question coming in. I'm not sure who the best person to ask this to is, so I'll ask you both, and maybe you can jump in. But how can assays be optimized for survivability of B cells?
Yeah, I can comment on that, and maybe Dina can add on as well. So I think what's important about the B cells, so I mentioned previously, it is a long duration where you have to leave the B cells on the beacon and be able to make sure that they're happy and viable. So what we do ahead of time is actually culture the B cells onto the chip with our media formulation, and then evaluate how they survive over time, even under the assay conditions that we're running. So it's really important. So we do pre-tests by fax just to confirm the robustness of the assay, but on the beacon itself, we have to do the assay optimization just to make sure the B cells are happy, and we can screen that later just by looking at IgG secretion over time and make sure that they are happy, that we can export them successfully and then recover the cDNA at the end. So it's a longer process to be able to confirm, especially if you're using reagents that might be a little bit different, that the B cells are not usually exposed to. There's certainly the importance of evaluating what that altogether looks like on the beacon is really important. So that's a great question.
Great. Looks like we have another one for Dina here. What is the efficiency of sequence recovery from B cells screened during the workflow?
Yeah, that's an important question, because once you have your B cells screened, I think the goal is to be able to recover them or capture their sequence. So we have a new release product called OptoSeq Plasma BCR, which allows you to essentially pull in mRNA capture beads that will go on to the respective pen, and it's going to do real-time lysis and then RT reverse transcriptase. Once your mRNA is captured on there, it will be stabilized cDNA. Then you can export those OptoSeq BCR beads. And we've shown, based on the slide that I presented earlier, we can get up to 85% solid recovery from multiple workflows.
Great. Shireen, what organic compartments can the beacon workflow work with?
So I believe the question is asking about the lymphoid organs, and so we've successfully isolated B cells from lymph node as well as bone marrow and spleen. We've also isolated the B cells from PBMCs as well for humans. So there's multiple lymphoid tissues that you can isolate the B cells from, and it really just depends on your immunization strategy, whether you're immunizing mice and what route of administration. That's really important to focus in on the lymphoid tissue that's most appropriate.
Great. Another one, Shireen, for you. Is enrichment of the B cell sample required?
Yes, definitely a key part of the process is to enrich the B cells. We've found that in addition to enriching the B cells, it's really important to know what the viability is. So you really want to put the enriched population with the highest viability on the platform so that you have more shots on goal. So when you're loading the cells into the pen, that what it's pulling is actually high viability enriched B cell population that you really care about. And that's when you can save time, because the process of loading takes a certain amount of time, and then culturing and then assaying. So you really want to start out with your pure population, high viability, and then you'll have a better chance of finding your hits at the end.
Great. Dina, we have another one for you. How many antibodies can be discovered from a single B cell sample?
So it really depends on the targets that you're working with, but in our validation work that we've done, working across 4 different chips and at running multiple workflows repeatedly, you can actually access, as high as over 5,000 hits to access the B cell repertoire. So just depending on the number of, depending on the targets that you're looking for and the number of viable plasma cells that you can screen on the chip. But our chip goes as high as a 14,000 scale chip that you can look at across, 4 different slots on the system.
Great. Shireen, here's another one. Can B cell discovery on the Beacon system screen human patient samples?
Yes, definitely. And we've initiated some of these studies as well. So we are initiating an effort with UCSF and the Gladstone, actually, to isolate antibodies from convalescent patient samples, patients who've recovered from the COVID-19. So that's certainly a process that's already in place, and there's isolation reagents that work quite effectively to enrich the B cells and put them onto the Beacon. So we're looking forward to the outcome of those studies.
Great. I see another one for Dina here. Is there a IgG quantitation performed, and are the binding blocking signals normalized to this?
So currently we are not able to quantify that signal yet, but there are lots of development projects where people are beginning to look at the different characteristics of that bloom signal that you see right above the pen. But, at the moment, if you're interested in looking at maybe understanding maybe how affinity works or some of those aspects, you can do some of those really unique assays that we've had customers do, where you will titer down different antigen concentrations and present them to your target antibody to see whether even at a lower antigen expression, it can still have strong binding. But that is something that we would continue to explore in the future.
Great.
Yeah, and
looks like we have time for about one more question. Oh, but go ahead. Were you going to comment?
Yeah, I was just going to add one more point. Thank you. So I think that's a really important question that gets at whether you can rank the signal on the Beacon, and having that ability to normalize, especially with the blocking or even the binding, would actually be a very powerful way to evaluate it further. With our blocking assay, though, what we found after looking at the purified antibodies was that because it was an image-based method for identifying blocking versus non-blocking activity, we really focused in on the most potent antibodies. And so we were actually able to pull out antibodies that had very consistent IC50s in terms of the blocking activity, and it had high potency in orthogonal functional assays later on. So we feel that having that image-based method up front adds a lot of value, even though at this point we're not able to normalize to the IgG secretion rate. But it's a really important question and something we're definitely working on as well.
Great. Let's take one last quick question and then we'll need to close. But here's the final question for Shireen. How do you test for robustness of your multiplex assay prior to harvesting immunized lymphoid tissue?
Yeah, that's really important. So there's a couple of factors, and that is to generate the reagents, evaluate them by facts, ensure that you have a really good MFI fold shift from your labeled reagents ahead of time before going on to the beacon. And I think another key component is having good control, positive control reagents to work with. Now, whether that's a hybridoma or a stable cell line that you can work with and just kind of load onto the beacon, get a sense of how that signal looks and whether you're able to differentiate between, for example, the multiplex assay human cyno mouse binding, being able to have that control and show that, you know, if I do have a cross-reactive antibody, that with each of these fluorophores, I'm able to differentially identify that I do have a true hit here. And again, starting from facts, starting with good reagents is key.
Perfect. Looks like that's all the time we have for questions today. So I'd like to thank Shireen and Dina for a great session, a lot of great questions. Just to remind everyone listening, the session was recorded. You'll receive a notification in 24 hours when the on-demand session is available for viewing. Before you log off, please take a moment to complete the feedback survey form on your screen so that we can continue to improve your digital week experience. And on behalf of Informa Connect Life Sciences, thanks for joining and have a great day.